And what we found is that, in fact, the behaviorally inhibited subjects showed significantly elevated activation bilaterally in the amygdala compared to non-inhibited subjects. That's basically a coronal slice of the brain showing you where the amygdala is, but you can see the actual changes in the bold signal, which is the level of oxygen, blood oxygen level, that is the signal that you're actually recording when a subject is in the fMRI scanner. And what you can see there is that for the behaviorally inhibited subjects, both left and right amygdala are more active in the contrast when they were looking at a fearful face and they were asked, how afraid are you of this face? Okay, so with that in mind, now that we see, by the way, go back for just a second, again, those are the subjects, the way we identify these behaviorally inhibited subjects is those are the subjects who we identified first at 4 months of age, but what I didn't mention is that in order for you to be characterized as an inhibited subject at 14 years of age, you had to be stable and high on observed behavioral inhibition at 1, 2, 4, and 7 years of age. So those are the stable, behaviorally inhibited subjects who are showing this heightened amygdala activation now at age 14. Okay. The next aspect of attention that we've examined is called attention bias to threat. And again, here's the same Ledoux cartoon, but now we've superimposed one other structure that I just want to mention here. The ventrolateral prefrontal cortex, you can see that up there on the right. And what you can see, there's the Ledoux cartoon up there. And basically, we are probing the circuitry of the response to threat in our behaviorally inhibited subjects with a dot probe attention task, which I'll describe for you in just a second. But the important thing to remember here is that the ventrolateral prefrontal cortex is thought to be a structure which downregulates the amygdala. So if we are looking at behaviorally inhibited subjects, we would expect that those behaviorally inhibited subjects who were quite anxious, they would show heightened amygdala activation, but little ventrolateral prefrontal cortex activation. On the other hand, if the subject was coping or dealing with the threat, we would expect them to show less amygdala activation and more ventrolateral prefrontal cortex activation. So we use a task called the visual probe task. This is a task which has been used extensively in the adult anxiety literature. And it's a pretty simple variant of a Posner spatial cueing task. Let me walk you through it. Subject's task is they see a cue, which is that cross right in the center, and then they have a target, and the target can be an arrow going down or an arrow going up, and their task simply is to press the right button if the arrow is going down, and the left button if the arrow is going up. Okay? Superimposed between the cue and the target, you put emotion faces. So in this instance, we have a neutral face and a face expressing anger. Okay? So this task was actually developed by a psychologist by the name of Colin MacLeod, who's at the University of Western Australia in Perth. And what MacLeod reported is that when anxious subjects, be they high in state anxiety on the Spielberger or clinically identified as being anxious, when anxious subjects were in this task and the target was congruent with the angry face, their reaction time was faster than non-anxious subjects. And when the target and the angry face were incongruent, they were actually slower to respond to the target. Okay, everybody get that? And so what you can do is you can compute a bias score, which is the difference between reaction time to the incongruent minus the congruent, and you can get something which McLeod described as attention bias to threat. There are literally hundreds of studies that have been done with this particular task with subjects who range from simple phobia to generalized anxiety to social anxiety to high state anxiety to high trait anxiety. Doesn't matter. The results are pretty consistent across different types of anxiety, that for those subjects who are anxious, you get this. You can see it here for high trait anxiety subjects, paper by Mogg and Bradley from 2004, that you get this attention bias to threat. They're faster when the target and the angry face are congruent. They're slower when the target and the angry face are incongruent. Okay? So we've actually done some work to see whether or not we can look at the underlying neural structures, as I showed you earlier, that are associated with performance on this dot probe task. So here's some imaging data, and what you can see here on the top right there is that when subjects are presented with very fast presentations of the angry faces, you get heightened amygdala activation. And in fact, that's a scatter. These are patients, adolescents with a confirmed diagnosis of anxiety disorder. What you see there is there's a positive correlation between the number of symptoms that the subjects report and the degree of amygdala activation in those subjects. However, when you present the faces for a long period of time, what you find is heightened ventrolateral prefrontal activation, and the degree of ventrolateral prefrontal activation is negatively correlated with anxiety symptoms. So the more anxiety symptoms, the less ventrolateral prefrontal activation. Again, with this notion that ventrolateral prefrontal cortex is associated with downregulation with the amygdala, and the amygdala is there to detect novelty or threat. We've done this attention bias task with our own behaviorally inhibited subjects. So here is, you can see there on the right, the high behaviorally inhibited subjects. By the way, we used both angry and happy faces. And what you could see there is that the high behaviorally inhibited subjects are showing a greater attention bias to threat compared to low behaviorally inhibited subjects. Interestingly enough, the low behaviorally inhibited subjects are actually showing attention bias towards happy faces as opposed to this bias towards threat. Okay, how about in terms of how this emerges and the learning processes that are associated with that? So if you think about anxiety disorders, there is a long tradition in the work in human anxiety about the role of conditioning or learning. And this actually links very closely with the neuroscience work that I told you about earlier, the Joe Ledoux's work and Michael Davis's work, which really used animal models, particularly rodent models, to look at fear conditioning as a way for understanding the development of or the emergence of anxiety or fear within rodent models and probing the neurocircuitry. In that literature, in the human literature on conditioning, the studies show that people with anxiety are unable to discriminate between ambiguous cues and overtly dangerous cues.